The gene editing technology CRISPR has been used for a variety of agricultural and public health purposes, from growing disease-resistant crops to more recently a diagnostic test for the virus that causes COVID-19. Now, a study involving fish that look nearly identical to the endangered delta smelt finds that CRISPR can be a conservation and resource management tool as well. The researchers think its ability to rapidly detect and differentiate among species could revolutionize environmental monitoring. The study, published in the journal Molecular Ecology Resources, was led by scientists at the University of California, Davis and the California Department of Water Resources in collaboration with the MIT Broad Institute. As a proof of concept, it found that CRISPR-based detectant platform SHERLOCK, that is specific high sensitivity enzymatic reporter unlocking, was able to genetically distinguish threatened fish species from similar looking non-native species in nearly real time with no need to extract DNA. The scientists focused on 3 fish species of management concern in the San Francisco estuary: the US threatened and California endangered delta smelt, the California threatened longfin smelt, and the non-native Wakaskai. These 3 species are notoriously difficult to visually identify, particularly in their younger stages. State and federal water pumping projects have to reduce water exports if enough endangered species like delta smelt or winter run Chinook salmon get sucked into the pumps. Rapid identification makes real-time decision making about water operations feasible. Typically, to accurately identify the species, researchers rub a swab over the fish to collect a mucus sample or take a fin clip for a tissue sample. Then they drive or ship it to a lab for a genetic identification test and awaits the results, not counting travel time that can take at best about 4 hours. SHERLOCK shortens this process from hours to minutes. Researchers can identify the species within about 20 minutes at remote locations non-invasively with no specialized lab equipment. Instead, they use either a handheld fluorescence reader or a flow strip that works much like a pregnancy test. A band on the strip shows if the target species is present. Anyone working anywhere could use this tool to quickly come up with a species identification. While the 3 fish species were the only animals tested for this study, the researchers expect the method could be used for other species, though more research is needed to confirm. If so, this sort of on-site real-time capability may be useful for confirming species at crime scenes, in the animal trade at border crossings, for monitoring poaching, and for other animal and human health applications.